All right. Hi, everyone. Today's lecture is our second one in our series of evolution. Remember, there's three of them all together, so there's one more to watch after this. But this is on tracing the evolutionary history of life. And so we're going to be basically following all the way from the oldest rocks all the way up to current time. Okay. So, bird feathers. That's where we're going to start. Bird feathers and flight are really a marriage in structure and in function. Birds were not the first feathered animals on Earth. Actually, dinosaurs were. Although I must say it's interesting because the oldest bird fossil that's ever been found was actually found in 2013, and it was found in northeastern China and is dated about 160 million years old. And this Archaeopteryx was actually the first fossil of record that had wings. We don't know how they got these wings, but they appear to be linked to dinosaurs. And scientists are still trying to figure this all out. They currently classify birds as dinosaurs. They used to be in their own phylum of aves, but now they're dinosaurs. And it kind of makes sense because if we look at leg structures and kind of this beak structure, there is a lot of similarities between, the scales on their legs and the scales on reptiles and so on. The evolution of birds, or in other words, the speciation of birds, is an example of what we call macroevolution. And macroevolution is looking at major changes that have been recorded in the history of life, and these have occurred over vast periods of time. Now, the Earth is estimated to be 4.6 billion years old. The oldest rock that has been dated was 3.5 billion years old. They know this by looking at various isotopes of carbon, and we'll talk about that a little bit more and how they do that. And these isotopes of carbon indicate that there was some kind of metabolic activity going on during this period of time. Now, if we were to think about, 4.6 billion years ago, can you even fathom what that actually means? Well, if Earth was 4.6 billion years old, and if we scale that down to representing 46 years, that means that humans have been here only 4 hours. That means the Industrial Revolution began a minute ago. And in that time, we have destroyed more than half the world's forests. I think that helps put things into a little bit more perspective, but still, this time scale is just really hard to fathom. The oldest fossils identified were the stromatolites, and basically what they are is they're banded domes of sedimentary rock in which there are layers of this cyanobacteria, which are basically blue-green algae or blue-green bacteria that were probably photosynthetic, although the earliest life forms were probably not photosynthetic. And why is that? Well, the reason is because the early atmosphere of Earth had very little oxygen. There was a lot of intense lightning going on. There was a lot of volcanic activity. There was meteorite bombardments occurring. UV radiation was very intense. Those early life conditions really, there's a couple hypotheses. One of them is that life may have evolved from 4 stages. The first stage is what is known as the abiotic synthesis of small organic molecules. Abiotic, of course, means without life, and synthesis means to take several of something and put them together to make something else. So these small organic molecules, like amino acids, those are the monomers of proteins that we know. These small organic molecules like nitrogenous bases that make up DNA and RNA. The joining of these monomers to make polymers like proteins and nucleic acids, then the packaging of those molecules into what we call protocells. Proto meaning first, so the first cells. And these led to the origin of the first self-replicating molecules that eventually made inheritance and the passing on of genetic traits possible. Okay, so what came first, the chicken or the egg? And a better way to put that is what came first, DNA or proteins. Okay, so let's try to answer that question. RNA, which you know is ribonucleic acid, acts like an enzyme, and we call it a ribozyme. And what happens is, as we know, RNA is made up of monomers of nucleotides called adenine, cytosine, guanine, and uracil. And those short little segments of RNA that make up these little segments of polymers can act like simple genes. And those genes can then be assembled together, and then they can be replicated. And so what we're doing is we're making copies of those RNA strands and the earliest first genes. And what were those genes coding for? Those genes were coding for proteins, RNA to proteins. And remember, those proteins are what makes you the way you are. It's what gives you those novelties. So genetic information made it possible to pass along the information for making other new molecules. So early on, it was RNA making proteins. Today, we have that, what we know of as the central dogma of biology, where DNA is being transcribed into RNA, which is then translated into proteins, which really helps protect our DNA and make sure that we're making an exact copy of it. Because remember, if we're making copies of RNA, RNA is not enclosed in a nucleus and therefore is subject to more changes or mutation in its segments. So, how did RNA form? Well, let's see. There are several hypotheses. One of these hypotheses is that the extreme heat and pressure that is put on some of the organic materials that are deep in our ocean could form inorganic materials. And some of the evidence we have for that is found down deep in the hydrothermal vents, which the hydrothermal vents are found deep in our ocean, and they follow along the mid-oceanic ridge. And this is where crust is being made. So we have hot mantle material being pushed up into the crust, and then it spreads out and it hits the cold ocean water, it solidifies, and we make more crust. Well, also in these ridge areas, we have, these are called hot spots or black smokers. And you can see on these black smokers, this orange or yellowish stuff that you see is really sulfurs. And these are sulfurs that have solidified into making these black smokers. We can see iron that's coming out of it, metal hydrides. These are substances that are under extreme pressure, they're under extreme temperatures, and these inorganic material that's being produced. Remember, to be organic, you have to have carbon, hydrogens, right? And the inorganics don't have that. So like iron, Fe, sulfur, S, and so on. So these inorganics are producing food, for example, for these other organisms, like these Riffia worms. They look kind of small in this picture, but they can actually get up to be about 6 feet tall and about a good foot around. And what you're looking at here is you're actually looking at the hemoglobin that is making up or within their modified gills. And and then these white things that you see are actually hard tubes that these worms are protected in, and they will extend out from these red pieces here, these modified gills, which are used for removing food from the water. And there is plenty of it available down there in the hydrothermal vents because of all the sulfur that's produced. These crabs actually look very small in this picture too, but they can get out to be actually about 3 feet across. And then these little polychaete type worms also can get to be, a foot long. They can get to be very, very big. And there's not a lot of competition down there because, again, it's very hot, it's very deep, there's no sunlight. So these guys are all undergoing a process called chemioosmosis. They are using chemicals for their metabolic processes. So there were a couple scientists that really wanted to research this and wanted to see if they could indeed make organic materials from inorganics. And the first two guys, Oparin and Haldane, in the 1920s, both of these guys worked independently in two different places. And they came up with a similar hypothesis or postulate in that the conditions on primitive Earth favored the abiotic synthesis of organic material. And as part of that, they said, Earth or the atmosphere of early Earth contained less oxygen than it does now. And because it was less oxidizing, there was no oxygen available. And as a result, they hypothesized that these inorganics could make organics. Now, Miller and Urey, about 30 years later, they really wanted to test Oparin and Haldane's hypothesis. And what they did in the laboratory was they created conditions that represented the atmosphere of early Earth, which was that there was water, there was hydrogen gas, there was methane, and there was ammonia. And they put this all together, added a spark to it, and lo and behold, they were able to create organic material from those elements. A number of other labs have done similar experiments, and they have managed to create all 20 amino acids. They've been able to create some sugars and some lipids, and they've been able to make all of the purine and the pyrimidine bases, the adenine, the guanine, the cytosine, and the thymine in a laboratory. And let's look at this short little podcast here of Miller's experiment so that we can get an idea of what they did.
Miller was intrigued by Oparin and Haldane's theory, so he designed an experiment to test it in the lab. His plan was to have a stab at reproducing the conditions of the early Earth, to see if anything related to life would form. It sounded so far-fetched that Miller's professor gave him just 6 months to produce a result, rather shorter than the millions of years the Earth had taken.
Now, this is the actual kit that Stanley Miller built. This lower sphere is filled with boiling water, and that represents the early oceans of the Earth. When Miller did the experiment, this top sphere was filled with gases that represented the atmosphere of the primordial Earth. That's methane, ammonia, and hydrogen. And these two probes introducing an electrical spark, and that simulated the violent, lightning-rich atmosphere of the early Earth, like this. Now, I can't do this experiment for real because it's actually highly dangerous. Unless you flush out all of the air in the system, the whole kit might explode. Plus, this electrical grid is live at 10,000 volts, so I really mustn't touch it.
Just 2 days into his experiment, Miller noticed that the water had turned pale yellow. After just a week, the flask was coated in an oily brown scum. Something weird was definitely happening in his primordial soup. But what was it?
Using a technique called paper chromatography, Miller analyzed the contents of the flask. And this is a copy of the actual results. Now, it may not look like much, But to Miller, it was absolutely sensational, because each one of these blobs is the chemical signature of a molecule called an amino acid.
So if you could imagine being in Miller's lab and discovering this, first of all, I can't even imagine if this guy won't even run the experiment. It sounds pretty darn dangerous, and that Miller was very lucky that he didn't electrocute or kill himself or blow up his lab. But you can imagine that if he's getting markings that match the markings of other amino acids, that would be a pretty exciting discovery. Okay, so macroevolution reflects major events that are occurring in the history or have occurred in the history of life on Earth. And what we're going to be doing over this next part of the semester is we are going to be working our way through these groups of organisms and looking at the evolutionary history of the novelties that exist in these organisms and how related they are to one another. So macroevolution, again, represents or reflects the major events in the history of life, and it's revealed in the fossil record. And it is going to, we're going to be looking at organisms at taxonomic groups that are higher than species. If you remember back earlier in the semester, we talked about classification of organisms, and we talked about how organisms are all separated into three domains. You have the bacteria, the archaebacteria, and the eukarya. Remember, you are a eukaryote because you have membrane-bound organelles and you have your DNA contained within a nucleus. But from domain, we go kingdom, phylum, class, order, family, genus, and species. So we are going to be looking at these taxonomic groups that are much higher than that specific level of species and determining how are they related to one another? What do they have in common? What novelties do they share? So we're going to be looking at those novel designs. We're going to be talking about how macroevolution produces explosive diversification in some groups. And it can cause mass extinctions, which will also change the diversity of life that we have on our planet. And then we're going to look at species selection. In other words, the species that endure the longest and produce the greatest number of new species are the ones that are going to determine the trends in evolutionary history. So the fossil record is really, really important. The fossil record is an ordered account of which fossils appear within which layers of sedimentary rock. Now, a lot of times people refer to the fossil record as being incomplete, like we don't know what's going on. However, we are constantly we are constantly finding new fossils. We just have to know where to look for them. Remember, trying to find a fossil is really difficult because, first of all, you have to consider the probability that one particular organism would die at one particular time in geographical history in which their body would be preserved. So either their body was preserved in ice, or their body was buried under volcanic ash, or there was some other event that froze their body in rock, or created an imprint of that body that was preserved in rock. Keep in mind that then not only is it the probability that any one particular animal would die at any one particular time during any one of these events that would help preserve the body, but what is the probability that that body would sustain or last through millions of years in these rocks and not be destroyed? And then what's the probability that any one person, any one scientist, anybody comes along and finds it? That's like finding a needle in a haystack that is, the width and length of the United States. Very slim, okay? And so we're constantly looking for fossils, and we're looking to fill in the record, okay? There's a film that I highly recommend you watch, and it's listed in your Canvas. It's called your inner fish, and there's another one called your inner reptile, and there's a third one that's called your inner monkey. And those are amazing videos, and they really talk about these fossil records, and that we have actually been able to find many of the links between organisms, for example, that lived in water and now live or then moved on to land. And those would be the tetrapods. So these fossils are imprints or remains. And keep in mind, when we talk remains, it's only the hard parts that withstand the passage of time here, like the bones, your teeth, your hair, your nails. Those are the only things that survive. Your soft body parts don't survive generally. They would have to be, frozen or something. And these fossils document the differences between past species and our present species. They also reveal that many of the species that have lived on Earth have actually become extinct. And so these fossil records reveal the historical sequence in which organisms have evolved, but that's only helpful if you actually know how old they are and where they fall in evolutionary history. And so talking about dating rocks and fossils, okay, in our next section, you're going to be watching a film in which these people are going to be looking for stickleback fish, and you're going to see how they have identified how old various layers or strata of the Earth, how old they are, and then from each of those strata, they're able to pull out fossils of fish that have been bedded in these rocks, and they're able to determine how old they are based on what is called relative dating. And relative dating, basically what we're doing is we're looking at the layers of rock. We know that the deeper the layers are, the older those fossils are going to be. The closer to the surface, the younger those fossils are going to be. Now, it doesn't really give us any idea of how old those layers are. If we were just to go out and hunt and look for them, we wouldn't be able to go, oh, look, that layer is light tan, so that means that they are 3.5 billion years old. Well, we can only maybe take a sample of that layer, and because it would be crazy to go and date every single organism fossil that you find in each layer. So they'll date, a representative sample, and then they'll look at all the organisms in that layer. And from there, they're using what's called relative dating to determine how old each of those layers are. If you've ever been up to the Dakotas or Wyoming, you'll know that as you drive along the road, there are markings all along the road that tell you you're passing through the Cambrian period, you're passing through the Precambrian, and they'll tell you how old those layers of rocks are right next to the freeway, which is pretty cool because it's like driving through millions of years worth of time. All right, so absolute dating is much more reliable in determining how old something is because we use what is called radiometric dating. And radiometric dating is based on the decay of radioactive isotopes, such as looking at carbon. And these are unstable forms of an element that can be used to date and determine the geological record of Earth's history. So let's say that this timer takes 1 minute for all of the sand to reach the bottom. If half of the sand has fallen through, how much time has passed? Well, if it takes 1 minute for all the sand to get to the bottom, and we know 1 minute is 60 seconds, then half of that would be 30 seconds, right? So if we look at this, let's say that this particular timer takes 4 minutes for all the sand to drop to the bottom. If 3/4 of the sand has fallen through, how much time has passed? Well, we know that 1/4 would be what? 1 minute. 2/4, which is a half, would be 2 minutes. We know that 3/4 would be 3 minutes because 4 minutes would be a whole, okay? So in this case, if we talk about this in terms of the Earth and how old the Earth is, we can use very similar principles to tell how much time has passed. If we know how much sand, for example, is going to pass through this timer, and we know how much has passed, we can determine how much time has passed. So we can do the same thing with our radioactive dating. Okay, so, oops, it repeated itself. All right, so let's talk about radioactive dating. This is a process then where unstable elements become stable. So for example, radioactive uranium, when it breaks down, it turns into lead, which is its stable form. Radioactive carbon turns into the stable form of nitrogen, and radioactive potassium turns into its stable form of argon. And so when we talk about it breaking down, that means it is decaying and it's turning from one element to another. Now, in terms of living organisms, we know how much carbon, or C14, now remember carbon is usually C12, and there's 3 actual radioisotopes, or there's 3 isotopes of carbon. And that means that they have a nucleotide that is different. So C13 has one more neutron, not nucleotide, neutron. So C13 has one more neutron than C12, and C14 has 2 more neutrons than C12. And so when those break down, we know what their half-life is. So for example, if we start with radioactive carbon, C14, we know that it takes 5,730 years for carbon to break halfway down. That means if we look at it and we say, okay, 25% of C14 remains, we know that we have passed 2 half-lives. So a half of 100 is 50, and a half of 50 is 25, while a half of 25 is 12.5. So if you think of this as 1 half year, this is 2 half years, this is 3 half years. So if we know 1 half year is 5,730 years, we can multiply, we know that that's 5,730. If we pass another half-life, so 50, half of 50 is 25, we know that we have gone 1 half-life, 2 half-lives, and we can take 5,730 and multiply it by 2, and you'll get 11,460 years. If we have 3 half-lives, we multiply 5,730 times 3, and we get 17,190 years. And that helps us determine how old these fossils are based on the decay, or how much carbon, or C14, is left in that organism after it has passed away. So here's another little look at radioactive dating or radiometric dating. Again, we know C14 has a half-life of 5,730 years old. What I like to do is I like to just set out myself a little timeline type thing. And I go, okay, if we start with 0, which would be my whole, and then we have a half of that, and then a half of a half is a fourth, and a half of a fourth is an eighth, and a half of an eighth is a 16th, and a half of a 16th is a 1/32nd, okay? So this would then represent my half-life, my 1. Then this would be my second half-life, my third half-life, my fourth half-life, and my fifth half-life. So what that means is that I would multiply this times 2. So if, for example, I say that this fossil contains 1/4 of C14 as it had when it was living, then I would literally take 1/4, and I go, okay, there's my 0, half of that, or my 1, half of that is a half of that is a fourth. This is my 1 half-life, 2 half-lives. That means I multiply 5,730 by 2, and I get 11.4. If it was 4 half-lives, if a fossil contained 1/16 of C14, 1, 2, 3, 4. So I go back and multiply 5,730 by 4, and I get about 22.8. Okay? So that's how you do radioactive dating. It's really not that hard. Again, I say the simplest thing is just to lay yourself out like a little chart, just draw a line, go 1, 1/2, 1/4, 1/8, 1/16, 1/32, and then just count the half-lives and then multiply by the number. That's the easiest way for you to figure out these problems. All right, so if we look at the geologic record of organisms, okay, and we start down here with the oldest known rocks on Earth, which are about 4,000 years old, or 4 million years. And we know that Earth is 4.6 million years old. Then we can look at the evolutionary trend that took place on Earth, where we had the earliest eukaryote cells appear, we had the earliest invertebrates appear, then we have vascular plants appearing, we got some fish appearing, we got ferns appearing, we got reptiles appearing, we got cone-bearing plants appearing, we got some dinosaurs appearing, we got flowering plants, and then each of these colors too represent major events in Earth's history. Like we know, in the Jurassic period, we had dinosaurs, and then Cretaceous period, that's when the dinosaurs became extinct at the end of that period. So it was a major event. And what ended up happening is we ended up having some new species that were able to move out into niches and survive and reproduce and really undergo some huge speciation events, okay, or development of new species. So that's what we are talking about at this moment in time. And then we will talk about these major speciation events and what happened during these events. Now, I'm not going to ask you to memorize any of the eras. I'm not going to ask you to memorize any of the periods or any of the epochs even. But we are going to start working our way through these organismal groups, and you're going to want to follow the trend as we cover them and look at the novelties that appear through descent with modifications. Okay, so this is it just in bigger picture for myself. And what we're going to do is I'm going to introduce you to some of the key ideas on that geological timeline or record that I was just showing you.
By studying various fossil records, geologists have put together a geologic record. Evidence in the fossil record of large-scale extinctions and/or enormous radiation of many diverse new life forms defines the main boundaries within the geologic record. As you move through the geologic record, the names and dates will change. Also, a diagram at the upper left will show what is known of the history of continental drift, based on evidence dating from the Mesozoic era. During the Precambrian time, which began at least 4,600 million years ago and ended 542 million years ago, life began on Earth. The first prokaryotic cells and cyanobacteria appeared, as well as the first animals. The beginning of the Paleozoic era, 542 million years ago, marked the origin of most modern animal phyla. The first plants, jawless fish, bony fish, and amphibians evolved. Amphibians became the dominant vertebrate life form, while the first reptiles and seed plants appeared. By the end of the era, 251 million years ago, many marine and terrestrial organisms had become extinct. The Mesozoic era stretched from 251 million to 65 million years ago. At the beginning of this era, the reptiles radiated out as dinosaurs to dominate the landscape, along with the cone-bearing plants, or gymnosperms. At the same time, the first birds and mammals originated. As the climate and Earth's surface changed, flowering plants appeared and many organisms, including most dinosaurs, became extinct.
The Cenozoic era followed the Mesozoic, 65 million years ago, and continues through modern times. The beginning of the era saw the radiation of the mammals, birds, and pollinating insects such as bees and butterflies. The angiosperms evolved to dominate the landscape as most modern mammalian orders appeared. The primate groups evolved, leading to the origin of ape-like human ancestors, and finally humans.
So, that leads into our next topic of discussion, which is looking at continental drift and plate tectonics as it relates to macroevolution. Now, continental drift is the idea that the continents move around the Earth's surface, and the surface of the Earth is broken into many little pieces, kind of like a jigsaw puzzle, and it's plate tectonics that actually describes how these pieces move on the Earth's surface. So seafloor analysis and other research actually supports what we know of as the hypothesis of plate tectonics. Now, it's kind of interesting because I know when I went to school, we got little pictures and they had the continents all drawn on them, and we were supposed to put them together as a puzzle. And I remember it was really difficult and we were having trouble putting them together. And then I remember our teachers talking about how that they had learned all this new information and that the continents don't really fit together. It's the plates themselves that fit together. So the plate boundaries are much larger than the continents themselves, and they extend out underneath the ocean. And once they gave us the new pictures to look at in terms of what the plates actually look like, things fit together in our puzzle much better. And I'm sure you guys have all done a similar type activity. Now, these crustal plates, as we know them, are constantly moving. We have continental crust that is running into other continental crust, and it's folding upwards and piling up and building up mountains. If you've ever been out on the 14 towards, let's see, Donner's Pass or heading to Magic Mountain, you can see out there where the Andreas Fault has, it's not actually the Andreas Fault. I can't remember the name of the fault, but it runs near the Andreas Fault. And you have these continental plates coming together, and they form these folds. And so when you look at the rocks, you see one on top of the other, forming these little mountainous type or huge mountainous type structures. If you ever get a chance to go out to Pear Blossom, you can hike all the way out to what is called the Chair. And it's quite a hike. The first part of the hike's not too bad. The last part of the hike is like switchbacks that are pretty steep. And then you get out to the Chair and you can overlook all of these plate boundaries that have caused this uplifting to occur. It's near the San Andreas Fault, but they are actually, I forget the name of them, but they have they're like the Pear Blossom Fault or something. They have a whole nother name. I apologize, I don't know their name right now, but anyway, we also have regions in which oceanic crust and continental crust are coming together. And as a result, what happens is the oceanic crust is a little bit more dense than the continental crust. And so what we end up with is the oceanic crust subducting underneath the continental crust in these areas called subduction zones. And in these subduction zones, we get trenches that form. We get hot spots that form volcanoes. We get other hot spots that form islands. I don't know if you realize this, but all islands, they form an arc because it takes it. Okay, so these volcanic islands don't just form overnight. They take years and years. Well, think about this. The globe is constantly rotating. And so as our globe is rotating, what we end up with is this arc of islands. They don't end up in a straight line. They end up in an arc. So if you look at the Hawaiian Islands or you look at the Archipelago Islands, they are all in an arc because of the rotation of Earth. So We also, here at that mid-oceanic ridge, this is where we have seafloor spreading. This is where that hot magma is coming up, and this is where we're going to find those black smokers and stuff as well. And we have crust being created, and it's moving out through these spreading centers, and it's going out to these subduction zones where that oceanic crust is being destroyed and being pushed back down into the mantle, okay? This area too is also where we're going to see a lot of earthquake activity because of those crusts running into each other. Now, just to make sure it's clear, remember the San Andreas Fault, those are faults in which the crusts are moving side by side from each other. Other crusts actually move right into each other, and that's what causes the folding up. All right, so if we go back and we talk a little bit more about plate tectonics, and we look at Pangaea about 250 million years ago, we know that that was a single land mass. There was a lot of volcanic activity, earthquake activity, and as a result, there were extinctions that occurred, and land masses started breaking up, which also caused this decrease in diversification of organisms as some of the organisms could not survive these changes in environment that were occurring as their continents moved to different parts as they broke up. And these land masses began breaking up about 180 million years ago, and they resulted in some geographic isolation that is referred to as allopatric speciation. Now, one of the ways I remember allopatric speciation is, I don't know how many of you have been to Hawaii, but when you go to Hawaii and you land, you get off the plane and they put this lei on you and they go, aloha. And so I kind of turn the language around and go, aloha, Patrick, for allopatric, just as a mnemonic device to help myself out. Because what allopatric speciation is it means there is a geographical boundary or border between species. And that separation of these species can lead to speciation or it can lead to extinction. So, for example, when the continents were connected together here, organisms could move from one continent to another. When they split apart and, part of Laurasia started moving up north, and we've got Antarctica moving down south and so on, we've got changing environmental conditions that are occurring, and then we're having these ocean masses that are separating the continents, which is our geographic barrier, which is creating this geographic isolation between species. And again, depending on whether the species has a trait that allows them to adapt to this changing environment or not, is going to depend upon whether they survive or they experience extinction. Okay, and so they're showing the continents spreading apart even further, and then back to where they are in present day and as we are familiar with them. So here's looking at a picture that shows these continents connected together, and it's kind of interesting because when we look at fossil records, we find fossil evidence of organisms. For example, this land reptile, we find fossils, or have found fossils in Africa, in India, and down in Antarctica. For this fern, we found them in Australia, Antarctica, India, Africa, South America. This freshwater reptile we found in Africa and South America. Same thing with this Triassic land reptile. Well, how do you explain that? The way that scientists explain it is that when these continents divided, these organisms became separated by geographical barriers, and they either survived this change in environment, or in the cases of many of these organisms, they became extinct. All right, so our continents are drifting apart about 2 centimeters a year, and eventually they will come together on the other side. These arrows represent the direction of movement of these various plates, and you can see some of them are diverging away from each other. Some of them are converging together, and some of them are sliding past each other, like up here on the North American plate. We can see it's pretty interesting because along the North American plate, and this, of course, is the other side of the Pacific Ocean, along the Philippine plate and the Australian plate, this is what's known of, and if we extend down here into South America, this right here is known of as the Ring of Fire, okay? And that Ring of Fire is plate boundaries that are around the edges of the continent in the Pacific Ocean, where we have those subduction zones and we have a lot of volcanic and earthquake activity. And if you pay attention to the news sometimes, it's kind of fun to predict. When you start hearing that there are earthquakes over here in Japan, you'll often notice that South America then gets a lot of earthquakes. Then you'll notice Central America gets some earthquakes. And you can usually predict that you're going to be somewhere in the line of things for having an earthquake shortly thereafter in that period of time that could be fairly significant. All right, so this here is a short little video clip on the evolution of continents, and we're going to go ahead and watch it.
The continents lie on massive plates that are in constant motion on Earth's surface. Their movement, called continental drift, sometimes forces continents together and other times pulls them apart. The continents as we know them today are still in motion, so millions of years in the future, their arrangement will look much different. Let's go back 540 million years to the beginning of the Cambrian period, when Earth's continents were mostly in the southern hemisphere and were coalescing into larger land masses. The largest, Gondwana, included the future South America, Africa, India, Australia, and Antarctica. Around this time, during a period of about 60 million years, a rapid diversification of life took place, known as the Cambrian explosion. Many of the major animal groups represented by species alive today first appeared during these evolutionary radiations. Over the next 200 million years, the large land masses continued to approach each other. At the end of the Ordovician period, massive glaciers formed over the southern continents, sea levels dropped about 50 meters, and ocean temperatures dropped. About 75% of all animal species became extinct, probably because of these major environmental changes. During the Silurian period, marine life rebounded, and the first vascular plants and terrestrial arthropods, scorpions and millipedes, evolved. Fishes diversified as bony armor gave way to the less rigid scales of modern fishes, and the first jawed fishes appeared. In the Devonian period, many animal groups radiated on land and in the sea. Fish diversified and some became formidable predators. Tall trees with fern-like leaves dominated newly appearing forests. The end of the Devonian is marked by a massive extinction of about 75% of all marine species. In the Carboniferous period, about 350 million years ago, extensive swamp forests grew on the tropical continents. The fossilized remains from the forests formed the coal we now mine for energy. The diversity of terrestrial animals increased greatly. Amniotes, with their well-protected eggs, evolved, as did giant amphibians and winged insects, the first animals to fly. During the Permian period, the continents merged into a single supercontinent called Pangaea. On land, the amniotes split into two lineages, the reptiles, and a second lineage that would eventually lead to the mammals. Toward the end of the Permian, conditions for life deteriorated. Massive volcanic eruptions produced ash and gases that blocked sunlight and cooled the climate. The resulting death and decay of forests rapidly used up atmospheric oxygen. In addition, much of Pangaea was located close to the South Pole by the end of the Permian. All of these factors combined to produce the most extensive continental glaciers since the snowball Earth hundreds of millions of years earlier. At the low oxygen concentrations at the end of the Permian period, most animals would not be able to survive at elevations above 500 meters. so about half of the land area would have been uninhabitable. Scientists estimate that about 96% of all multicellular species became extinct. The few organisms that survived the Permian mass extinction found themselves in a relatively empty world. In the Mesozoic era, atmospheric oxygen concentrations gradually rose. Pangaea remained largely intact through the Triassic period. On land, conifers became dominant plants, and frogs and reptiles began to diversify. The radiations of reptiles eventually gave rise to crocodilians, dinosaurs, and birds. The first mammals appeared during the Triassic. The end of the Triassic was marked by a mass extinction that eliminated about 65% of the species on Earth. Pangaea began to break up, and by the late Jurassic period, Pangaea became fully divided into two large continents, Laurasia, which drifted northward, and Gondwana to the south. Most of the large terrestrial predators and herbivores of the period were dinosaurs. The earliest known fossils of flowering plants are from late in this period. During the Cretaceous period, Laurasia and Gondwana broke apart. During the Cretaceous period, Earth was warm and humid. Flowering plants began the diversification that led to their current dominance of the land. By the end of the Cretaceous, many radiations of animal groups on both land and sea had occurred. A meteorite caused a mass extinction at the end of the Cretaceous. On land, almost all animals larger than about 25 kilograms in body weight, including the non-avian dinosaurs, became extinct. Several significant continental collisions occurred during the Tertiary period. By about 35 million years ago, the Indian plate ran fully into the Eurasian plate, and the Himalayas began to be pushed up as a result. Africa subsequently collided with Eurasia, and South America with North America. In this period, flowering plants dominated on land, and a rapid radiation of mammals occurred. Although the early Tertiary was hot and humid, Earth's climate began to cool, and grasslands spread over much of Earth. We are living in the Quaternary period, during which 4 major and about 20 minor ice ages have occurred. During ice ages, massive glaciers spread across the continents, and the ranges of animal and plant populations shifted toward the equator. The last of these glaciers retreated from temperate latitudes less than 15,000 years ago. Organisms are still adjusting to this change. Many high-latitude ecological communities have occupied their current locations for no more than a few thousand years. It was during the Quaternary period that divergence within one group of mammals, the primates, resulted in the evolution of the hominoid lineage, eventually leading to our modern human species, Homo sapiens. It may be hard to visualize, but humans are incredibly recent arrivals on this 4.5 billion-year-old planet. Even the Cambrian explosion, at about 500 million years ago, could be considered a recent series of events. Life has been evolving on Earth for about 3.8 billion years. The continents in Earth's biological communities have been constantly changing during this time, and will continue to change into the future.
Can see that is a lot of changes. That is a lot of extinctions and radiations and changes that have taken place, with 5 mass extinctions occurring over the last 50 or 500 million years, with a 6th one on the horizon. So therefore, over 50% of the Earth species have gone extinct. And again, most of the attention is given to the Permian extinction due to a lot of volcanic activity. And then a lot of attention is given to that Cretaceous extinction with the impact of that asteroid and the extinction of the dinosaurs. Now, the 6th mass extinction that is predicted is called the Holocene extinction. And the prediction is that there's going to be a loss of current species at a critical risk of extinction. We're not sure what it's going to be due to. It could be due to human neglect and interaction. It could be due to a coronavirus. It could be due to pollution, any number of events. It could be a geographic or geologic disaster that occurs. Once a mass extinction occurs, it takes about 5 to 10 million years for diversity to return to its previous levels. So what are all these radiations that we're talking about? And these, how these adaptive radiations create novelties that increase the diversity of organisms on the Earth. Well, during periods of extreme change, what happens is, okay, I like to give my cartoon version. Okay, so bear with me. Let's say that you're a small little rodent, and you are during the dinosaur period of time, and you're used to kind of like skirting along and going out at nighttime to get your food and hiding from these dinosaurs. And then one day this asteroid hits Earth, and all the dinosaurs are belly up, and you look outside and you're like, whoa, what's going on? And the next thing you know, it's all quiet out there. And so you get up one morning and you venture out, and you find out you got all the way to your, happy feeding ground, and nobody ate you. And you run back home, and you tell, Wilma, hey, Wilma, guess what? I made it all the way to blah, 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 and I didn't get eaten. She goes, you silly man, you. Anyway, he goes, well, let's all go tomorrow as a family. And so he packs up the kids and packs up Wilma, and they head on out to their favorite feeding ground, and no dinosaurs. And they go, So Fred says, you know what, Wilma, tomorrow I'm going to go out further and I'm going to see what I find. And so he gets up and he heads out, and he finds this beautiful area with a beautiful waterfall, lots of trees, a lake for the kids to swim in, and everything he ever imagined he would need or want in life is in this area. So he goes home, packs up the family, and they move. And now they are moving into an area that was previously inhabited by something that might eat them, maybe a dinosaur. And so what's happened is now they are moving into a niche that was previously unavailable to them, and they are able to exploit that area and use it. And what ends up happening is other organisms that have moved into that area of the same species, they start interbreeding. And as a result of those breedings, what happens is that group, their genes become different, or they develop a gene pool that is different than the gene pool from their original population. And as a result, speciation may take place, in which now Wilma and Fred's great-grandchildren, great-great-great-grandchildren, whatever, down the line there are an entirely new species. And so that's what can happen due to these adaptive radiations. And these adaptive radiations really do oftentimes follow the colonization of new unexploited areas, or they may result because of the appearance of evolutionary innovations, changes that maybe this population has that allows them to adapt to the change in the environment, such as those mutations of the antifreeze gene in the change in the environment as Antarctica moved and the water became extremely cold. This was an area where nobody lived before. And so this was a new unexploited area altogether, and the white-blooded fish were able to adapt and survive in that environment, and as a result, we have a radiation that has occurred. All right, so what's an evolutionary novelty? This is something that makes you different than groups that, or your ancestors that came before you. So, for example, wings in a bird are novelties. Eggs with shells are novelties. Amniotic eggs are novelties. Four limbs are novelties. Bony skeletons, vertebrae are all novelties. Now remember again, this is a evolutionary tree, oftentimes referred to as a phylogenetic tree. This would be back here, the ancestors to everybody to the right. This would be a common ancestor to the sharks, and everybody to the right. This would be a common ancestor for amphibians, and everybody to the right, and so on. And remember, it's these derived characteristics or novelties that help us to determine the evolutionary relationships of these organisms. So these novelties are going to, this goes back to descent with modification. So these novelties are going to evolve by gradual changes that are occurring in existing structures. And these existing structures may be co-opted to perform different functions. And if you remember back to genetics, it's the genes that are within your chromosomes that are controlling development. It's the genes that are playing a major role in the development of these evolutionary novelties that may change the rate or the sequence of development, and they may change the shape of the adult form or the basic body design of a particular organism. And if it changes into something that best allows it to adapt, survive, reproduce in that environment, then it will. And so that takes us back to those homologous structures. Remember, we have structures that have similar, well, they're the same bones, but they have been modified according to their function, all right? And then this here is, I know I talk about this more in your next lecture, but this is just looking again at some of those modifications that can occur from throughout time as organisms become more complex. And so a limpet is a fairly simple organism. It has one little shell, it's got a snail-like body. It's got an open circulatory system, and then we move on up to a squid who has a closed circulatory system, able to move very fast, chase after predators, escape prey. And then this is just looking at the eye receptors and how the simplest organisms have basically just pigmented photoreceptors to determine light and darkness, and then those eyes become more complex over time. And again, I'll talk about this in more detail in the next lecture. All right, so let's look at some mechanisms for macroevolution.
Microevolution by itself does not explain the diversity of life on Earth today. Let's investigate the role that macroevolution plays in this process. Multiplication of species creates biological diversity with the evolution of many different species from one common ancestor. This can be seen with Darwin's finches in the Galapagos Islands. Sometimes referred to as branching evolution or adaptive radiation, this occurs when multiple habitats and niches open up with relatively little competition. Evolutionary novelty results from the gradual refinement of existing structures for new functions. Microevolution by itself does not explain the diversity of life on Earth today. Let's investigate the role that macroevolution plays in this process. Multiplication of species creates biological diversity with the evolution of many different species from one common ancestor. This can be seen with Darwin's finches in the Galapagos Islands. Sometimes referred to as branching evolution or adaptive radiation, this occurs when multiple habitats and niches open up with relatively little competition. Evolutionary novelty results from the gradual refinement of existing structures for new functions. Here we see how ancestral birds may have used wing-like forelimbs for courtship. They could also have taken advantage of these enlarged forelimbs to allow for extended hops and gliding ability. Once flight itself became an advantage, natural selection would have remodeled feathers and wings to better fit their additional function. Explosive diversification often occurs following the evolution of an important adaptation. For instance, the evolution of the flower from early seed plants that were similar to modern conifers allowed plants to reproduce more efficiently and thus exploit many previously unavailable habitats and niches. After a mass extinction, previously filled niches and habitats are opened up. When the dinosaurs became extinct, mammals had the opportunity to evolve into those niches and produce incredible diversification.
remember, evolutionary trends does not mean evolution is goal-directed. Fossil record seems to show trends in evolution of many species, and it may be the result of species selection or natural selection in changing environments. So if we talk a little bit more about taxonomy, taxonomy is the branch of biology that's concerned with naming and classifying organisms. And if you think back all the way to one of our first lab activities, we did something called the dichotomous key, where we looked at two different structures and we had either or choices. They were tall or they were short. If they were short, you went to choice B. If they were tall, you went to choice 3, and you read choices all the way down. And what we were trying to do was we were trying to identify organisms down to their most specific category, which was species. We talked about how we can divide organisms into 3 domains. You could either be in domain bacteria, you could be in archaebacteria, or you could be in eukarya. And you guys are all eukaryotes because you have membrane-bound organelles and you have a nucleus that surrounds all of your DNA material. And then we can break the domain eukarya into kingdoms. Remember, we got the plants, the animals, the protista, and the fungi. And then we can further break those down into phylum, class, order, family, genus, species. Keeping in mind, genus would be like your first name, species would be like your last name. So if I talked about John going to the store and doing something horrible, you might be going, first of all, what did John do? Second of all, you might be going, John who? And it's the who part that gives us that species name or that specific name that tells us what John is that we're talking about. Okay. So that is what's referred to as binomial nomenclature, or 2 names. And taxonomists try to assign species into these 2 names so that we specifically know who we're talking about. And remember, these 2 names, the genus species name, the genus name is capitalized, the species name is not capitalized, but both the genus and the species name are italicized. And they are normally in Latin, and they are normally universally named, so that if I talk about Panthera pardus in the United States, then somebody in Africa knows that I'm talking about Panthera pardus or this cheetah, no matter where I'm at in the world, okay? So these are the most specific categories. The genus, species, the broadest category is the domains. Make sure you have those memorized: domain, kingdom, phylum, class, order, family, genus, species, okay? So let's look at some classification schemes to help you out a little bit more.
In the early days of biology, all organisms were classified as either plants or animals. Single-celled eukaryotic organisms such as algae and paramecium were assigned to one of the kingdoms according to whether they are photosynthetic or not. Fungi and prokaryotes were grouped with plants because fungi are sedentary and prokaryotes have cell walls. A 5-kingdom scheme was devised because some organisms don't fit well into a 2-kingdom system of classification. The simple cells of prokaryotes set them apart in their own kingdom, Monera. Organisms in the other 4 kingdoms all have eukaryotic cells. Fungi are not photosynthetic, so they are given their own kingdom. The kingdom Protista is an assortment of eukaryotes that don't fit into the other kingdoms, mostly unicellular organisms such as paramecium, unicellular algae, and multicellular algae. The 5-kingdom scheme divides living things into 2 fundamentally different groups, prokaryotes and eukaryotes. New data suggests that 2 kinds of prokaryotes diverged early in the history of life, and thus there are actually 3 major categories of organisms. A new classification system assigns more significance to the ancient split by creating a level of classification larger than a kingdom, a new category called a domain. The 2 domains of prokaryotes are bacteria and archaea. The domain eukarya encompasses all of the kingdoms of eukaryotes. Taxonomists are in the process of sorting out various kingdoms of prokaryotes within the domains bacteria and archaea, and also splitting the protists into several kingdoms within the domain eukarya.
All right, so when we talk about the classification of organisms, we can also look at their phylogeny and we can look at their systematics. Phylogeny, the goal of classification is to reflect the evolutionary history of a species or group of related species. So when we talk phylogeny, I've been mentioning a couple times about phylogenetic trees. And with those phylogenetic trees, we are looking at how they relate in terms of evolutionary history. Systematics is the study of biological diversity in an evolutionary context, which includes taxonomy, which includes the classification of organisms and determining those evolutionary relationships based upon scientific evidence. Okay. So again, if we go back and we look at this phylogenetic tree of carnivora, we can read this as this is where all of these carnivore common ancestor is. And then this is at the order level. So remember, we have domain, kingdom, phylum, order, family, genus, species. Oh, I missed one. Kingdom, phylum, class, order, family, genus, species. And so we're looking at the order is carnivora. The family, notice that even though they were in the same order, that their family name is different, which means they have a novelty or a trait that makes them different than the other two categories. And then we head on down to genus, and we see that we now have 4 genuses from those 3 families. And those genus names, again, are like their first name. And then we get down to the species name, which is their specific name. So for example, if we look at the coyote and the wolf there, we can see that their genus name, they are both Canis. And then their species name, we have specifically a wolf, which is latrans. And then we have the Canis lupus, which means the wolf. And again, notice that the genus name is capitalized, the species name is not, but they are both italicized. All right, so evidence of common ancestry. Remember, we've been looking at homologous structures, so we're looking at similarities in structure. We can also look at molecular sequences as providing evidence of this common ancestry used to determine evolutionary relationships, or what's called phylogeny. Some similarities are due to adaptations in organisms from different evolutionary lineages, and that process is what is called convergent evolution. So if we look down here at divergent evolution, they've come from the same common ancestor and they've diverged out, kind of like the Canis, the wolf and the coyote, okay? On the other hand, when we look at convergent evolution, they have different ancestral relationships, and they have converged together in terms of functions. So for example, and we also refer to this convergent evolution as analogy. So analogy without similarity. So for example, if you have a bat that flies and you have a butterfly that flies, they're coming from 2 different lineages, but they both fly, okay? And then the last one here would be 2 different lineages, and they don't get any kind of traits that are similar in function or structure. Now keep in mind, the structure of a wing on a bat is very different from the structure of a wing on a butterfly, okay? All right, so cladistics is grouping organisms by common ancestry. And when we're making these phylogenetic trees, each of these branches are referred to as a clade. And what it does is it allows us to look at the ancestral species and the evolutionary relationships of these species. In this case, we have basically one ancestor that gives rise to everybody. But in this case, we have this ancestor gives rise to this, and there's this divergence here. And then this ancestor here gives rise to this ancestor, but here's our divergence, okay? We're not going to worry about monophyletic or polyphyletic at this point in time. It's beyond the scope of what we're studying. Went the wrong way. So cladistics is based on the Darwinian concept of descent with modification from a common ancestor. And keep in mind, there's two types of characteristics here. You can have a shared ancestral characteristic, which means that it is common to a particular clade or a group of members in that clade, but it originated from an ancestor that was not a member of that clade. So this particular ancestor did not have the trait that is shared between these two individuals. And but remember, they have everybody after them has that trait. If we talk about this shared characteristic, nobody before them had it. Everybody to the right or above them has that trait. So a shared ancestral character is common to members of that clade, but a shared derived trait is only common to members of the clade that's above it in this case, or to the right of it, and it is not found in the ancestors. Okay, and we've gone over that a couple times. So again, if you have any questions or you're confused, please make sure you ask, okay? So shared characteristics can be used to construct these phylogenetic trees. Parsimony is the adoption of the simplest explanation for these observed phenomena. And it's what we oftentimes use to make things simpler and less confusing. So when we look at this, for example, if we look at red feathers, and let's say that we're looking at the evolution of red feathers in a species. If we put that red feathers evolved here and red feathers evolved here, it just makes the data more confusing than if we show the shared or derived characteristic occurred here and both of these have red feathers. If this one, for example, had something that was different than this one, maybe the shape of beak is different, or maybe they have a coloration that is different, or they have, feathers on their beak and this one doesn't. What we would do then is we would record that characteristic here that shows that this one has a characteristic that's different than this other red feathered one. But to keep things simple, this one is simpler and more parsimonious than this one here, okay? So the simplest explanation is often the best explanation. When we look at the genome and the molecular biology of organisms, the more recently two species have branched from a common ancestor, the more similar their DNA sequences are going to be. And we can use molecular systematics. In other words, we can use DNA or other molecules to infer the relatedness of one species to another, okay? And if we were to, for example, look at amino acid sequences in this cytochrome C protein, these are all the amino acids that are found in a human. And what we can do is we can compare these amino acid sequences from one species to another, and we can look at similarities between amino acids from one species to another. So for example, if we look at amino acid 11, we see that a pig, a chicken, a dogfish, and Drosophila all have valine in the same order. And then in number 12, they have glycine, 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 glycine. And in number 15, they have alanine, 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 which suggests that a pig, a dogfish, and a Drosophila are more closely related than wheat or chickens. And anyway, in my lecture on speciation, I'm going to go into this in more detail, and we're going to look at this a little bit closer. So homologous genes are found across distantly related species. And it's interesting because when we look at this molecular biology data, it demonstrates that all living organisms share many biochemical and developmental pathways, and it provides overwhelming support for Darwin's theory of descent with modification. Now, keep in mind, Darwin did not have access to any of this information or any of this technology that we do now. And so I think if Darwin was here today, he would just be blown away by how much evidence we have collected and how it supports his ideas of descent with modification and his theory of natural selection. And so this is just another example of a phylogenetic tree that's in your book. And I think I've talked a lot about phylogenetic trees, so I'm going to just move on. We've already talked about those 5 original kingdoms. We've talked about the splitting up now of those 3 domains and then the kingdoms within those domains. So make sure you know them, okay? And then we've gone over the common ancestor. And like I said, we're going to be working our way through these various kingdoms and talking about some of the species within these kingdoms. So at this point in time, you should be able to describe the conditions on the surface of early Earth. You should be able to describe evidence that life on Earth existed 3.5 billion years ago. You should be able to describe the 4 stages that might have produced the first cells on Earth. You should be able to talk about Stanley Miller's experiment in understanding how life might have first evolved on Earth. You should be able to describe the significance of protocells and ribozymes. You should be able to explain the key events in the history of life on Earth, explain the difference of radiometric dating and relative dating. You should be able to describe the theory of life or history of life on Earth and the key events that serve to divide those eras. You should be able to describe how Earth's continents have changed over the past 250 million years and the consequences that those changes had on life on Earth. Explain how volcanoes and earthquakes result from plate tectonics. Describe the causes, the frequencies, and the consequences of mass extinctions over the last 500 million years. Explain how and why adaptive radiations occur. Explain how genes that program the development function in the evolution of life. And then explain why evolutionary trends do not reflect directions or goals. Distinguish between homologous and analogous structures and provide examples of each, and describe the process of convergent evolution. Describe the goals of systematics. List progressively broader categories. That's that domain, kingdom, phylum, class, order, family, genus, species, all the way down to that most specific species level. Define the terms clade, derived characteristic, shared ancestral characteristics. Don't worry about in-group, out-group. You should know what a phylogenetic tree is. You should know what parsimony is. You should know how molecular biology can be used as a tool in systematics. You should explain how molecular clocks are used to track evolutionary time, and why a diagram of a tree of life is difficult to construct. And then a reminder just to consider all cells come from old cells. These cells are not created from scratch. If we broke 1 billion into 1,000 million, then consider that the number of seconds in a year is 31,557,600. So you will live 1 billionth of your second of life at age 31 and 8 months. Therefore, 1 billion seconds equals 31.8 years. So if you spent $1 million a year, you would spend $2,739.73 a day. So if you were to spend $1 billion in a year, you would need to spend $2,739,730 every day that year. So once again, a time scale is very difficult to fathom. And so sometimes we use these analogies to help make it a little bit easier and to understand the vastness of this time scale. And that's it for this part of your lecture. Make sure you watch the other 2 parts as well. And again, if you have any questions, contact me, let me know, and we will talk to you soon. Bye.